New Brunswick this morning. Thank you so much, Dr. Redmond. Whenever you're ready, you can go ahead.
Okay, great. Thanks, Jennifer. Can you hear me okay?
You're coming through loud and clear, and you'll be speaking to us today on precision oncology.
Great, and you can see my slides.
Absolutely.
Okay, well, thanks again. Thank you for the kind introduction and to you and SEPTAP and the others who've organized this meeting for inviting me to speak to you today about precision oncology. My name is Tony REIMAN. I'm a physician. I'm a medical oncologist. So I have spent the last 25 years treating cancer in the clinical setting, and I'm also a cancer researcher and affiliated with the University of New Brunswick and Dalhousie University, and my practice is in St. John, New Brunswick at the Cancer Center at the St. John Regional Hospital. And together with Dr. Sherry Christian and Dr. Robin Urquhart, it's my privilege to basically try and help shepherd the Atlantic Cancer Consortium as one of the co-leads for the consortium. I've been asked to talk to you about precision oncology. So I'm going to spend the next little while doing that. Let's see if I can get my slides to advance. Oh, there we go. So I thought I would start just by telling you about a patient of mine who sort of illustrates some of the promise and some of the work yet to be done in precision oncology. And we'll talk through the course of this. I'll talk through the course of this slide deck about what precision oncology is along the way, and some of the things that the Atlantic Cancer Consortium is doing to try to advance precision oncology in our region and together with our colleagues across the country. So this is the case of Mr. Smith. That's not his real name, but when I met him, he was a 54-year-old man. He's a farmer, and he first was brought to medical attention because his wife noticed a mole on his chest that was changing. And these aren't pictures of him, but his mole would have looked something like this on the left panel, this brown irregular mole that was growing and changing. And so this was biopsied, and again, not his biopsy, but under the microscope, it would have looked something like this panel on the right. This is a melanoma, a form of skin cancer. So this gentleman was referred to a surgeon who removed the melanoma. He went on to see me, and together with his surgeon, we did some further evaluation to see if there was any sign that this melanoma was contained and whether it was completely removed, and it appeared based on our investigations that no other cancer was found at that time in his body. So we told him he didn't require any further therapy, and we advised him that about 3 out of 4 people who present with a melanoma like his are alive and free of recurrence of their melanoma in the long term. And so we were hoping he would be in the majority, and wished him well and sent him on his way. Unfortunately, he came back to us 4 years later, referred back by his family physician who'd been following him since his diagnosis for signs of recurrence. And unfortunately, Mr. Smith developed widespread pain in his bones and became bedridden. He wasn't able to work. So we saw him again. He had a CT scan. This is one of his CT scans. You can see with the red circle, it's showing you a tumor in one of his ribs. This is a CT scan of his chest. It's a cross-section of his chest. It just gives you one example of the cancer that had spread throughout his bones. So this was what was giving him his pain, and he had a biopsy of one of these bone abnormalities. Since it had been so long, 4 years since he'd had his melanoma removed, he wanted to see if this was his melanoma or something else, and this turned out to be his melanoma that had recurred and had spread to his bones as well as his liver and lungs. So at that point, we turned to precision oncology to try to figure out what to do for him. So the biopsy material that had been obtained was sent to our hospital laboratory, where DNA sequencing of the tumor was undertaken using machines, something like the one on the left. What was found was a mutation in a gene called BRAF, or BRAF, in his tumor. And what's shown on the right-hand panel is sort of a stylized image of a cell showing some of the molecular consequences of this mutation. So the feeling was that this BRAF mutation was driving the behavior of his cancer. This gene BRAF encodes a protein that is overactive in these cells and is driving the cancer to behave like a cancer. So this was insightful and also had some implications for how we could treat him. The reason we were looking for this type of mutation is that this gentleman's cancer could be treated with therapy targeted to block the effects of this BRAF mutation in his cancer. So he was given a combination of 2 pills. One is a BRAF inhibitor, another one is something called a MEK inhibitor. These 2 things can work together to block the effects of an overactive RAF protein encoded by this BRAF mutation. And so he was prescribed this therapy, which is 2 pills that he could take at home. And this worked really well for him in the short term. This gentleman who was bedridden when he received this therapy took this treatment, and within a short time his pain resolved, and within a couple of months he was back at it. No time to waste. Had a farm to run and got back to running his farm. This treatment contained his cancer for a couple of years. Unfortunately, his cancer progressed after that, and he required further therapy, which was able to contain his cancer for another few years. So he was able to live for about 5 years after the development of this recurrence of his melanoma, and to keep his farm going. So this is an illustration of both some of the promise and some of the limitations of precision oncology today. This is the idea that if you can understand the biology of a cancer in an individual patient, that perhaps you can tailor treatment to that biology and achieve a good result for the patient. At the same time, clearly we did not cure this gentleman with our precision oncology approach with this targeted therapy, but he was able to benefit from the treatment for a while. So there's clearly still more work to be done here to improve the outcomes for gentlemen like Mr. Smith. For those of you who are, maybe not well studied on what cancer is, I thought this might be a good opportunity just to talk briefly about that, understanding that this is a conference aimed at patients and people living with cancer and their families and their loved ones, and maybe you haven't been spending your life studying this topic like I have. What is cancer? This is a picture of a lung with a big tumor in it. So the reddish part is the lung. It doesn't look perfectly normal. It's been damaged by years of cigarette smoking in this case, but there's this big white round tumor in the lung, and this is a lung cancer. This is what we would call a primary cancer of the lung, because this cancer has arisen from within the lung. The lung is where this cancer started, and this tumor has grown within the lung and has grown to a good size and is invading the surrounding lung tissue. So this is what a cancer looks like, in the flesh, so to speak. Here's an example of a liver from someone who had breast cancer. Originally, they had a cancer removed from the breast, which was the primary tumor. The site where the cancer started was the breast. But in this case, there were tumors in the liver as well. And so looking under the microscope at these tumors, you will see that they resemble the cancer from the breast. and that this represents spread of breast cancer from the breast to the liver, rather than a new cancer arising in the liver. Okay, so from these observations, we learn that cancer cells spread through the body. This is a cartoon showing this. The blue little round blobs are cancer cells. As you know, our bodies are made up of trillions of cells. You can fit about 1 billion of them on the head of a pin. And tumors, cancers, are also made up of cells. These cells originally were derived from our normal healthy cells, but these cells have become cancer cells. And what this picture shows is these blue cancer cells getting out of the area where the cancer started and into a blood vessel, which is that pink sort of tube in the middle. These cells can then circulate through the bloodstream, exit the bloodstream at some site distant from the tumor, as shown down below on the bottom half of this picture. These cells can get out of the blood vessel and into a new secondary site, like the liver, for example. And then these cells can subsequently grow into tumors in that secondary site. And so this is what had happened with Mr. Smith and with the person whose liver I showed you a moment ago. This is an example of a scan from one of my patients showing most of their body, kind of from the neck down, and all the orange or orange scale stuff, the bright orange stuff. Most of that is cancer that has spread through this person's body. This is a PET CT scan. And so this is an example of how we can examine the extent to which a cancer has spread through the body like this. And this process is called metastasis. And these tumor deposits in other parts of the body are called metastases. So these are some of the essential features of cancer. And, one question that arises then is, how does this happen? So as I mentioned earlier, it's thought that a given cancer arises from a single cell in someone's body that starts as a normal cell, but eventually transforms or turns into a cancer cell. And the mechanism by which cells transform it seems most of the time, and primarily, that the mechanism is that there are mutations that are acquired by that cell in its DNA. So changes to the genetic code of that cell are acquired at some point in the life of that cell, mutations in the DNA that the cell does not repair. The cell manages to survive this damage, and this DNA damage gives this cell cancer properties, which include the ability for that cell to replicate itself, or to multiply, or to proliferate. These are some of the words we would use to describe how one cancer cell can turn into multiple cancer cells by reproducing itself, which is something that healthy cells can do too, but this starts to happen out of control. And this is how a single cancer cell turns into a tumor. And then these individual cells can potentially spread through the body to form new tumors elsewhere. Okay, so this is one of the fundamental ways that cancer seems to arise. So there's lots of evidence now that DNA mutations drive cancer. I just want to emphasize that most of these mutations are not inherited, but rather are acquired by these cells during their life. So we are not necessarily born with these mutations. Most of the time we're not. In 5 or 10% of cancers, though, some of the mutations found in a cancer are ones that were inherited and have given an inherited predisposition or susceptibility to cancer. But most of the time that doesn't seem to be the case, and these mutations are acquired. Our cells are constantly experiencing damage to DNA. It's estimated that about 10,000 DNA damage events occur to every cell every day in our body. And our bodies have an extraordinary ability to either repair that damage, and carry on, or if a cell has damage that can't be repaired, there are mechanisms by which these cells, are destroyed. Some cells will self-destruct, others will be cleared by our immune system, and they don't become cancer. But if a cell is able to survive this and the wrong mutations happen to the wrong genes, then this DNA damage can lead to cancer. Okay, like in the case of Mr. Smith. who had cells that had this BRAF mutation that I showed you earlier. So, this understanding has come about over time, and there have been some landmark scientific advances that have enabled us to consider precision oncology as an approach to cancer care. One was in the late 20th century when the first human genome sequences were generated. This was a huge amount of work by many people over many years that cost a huge amount of money, using older techniques to examine the genetic code and sequence as much of it as we could. That's a lot of DNA. So that was a landmark advance, and this advance allowed us to get much quicker at sequencing future genomes. Once we'd gotten one down, we started to figure out how to do many more quickly from there. One of the methods for doing that is called next generation sequencing. So the original sequencing of the human genome was done with older methods that could only sequence one strand of DNA at a time. Now we have next generation sequencing approaches which are able to sequence millions of pieces, segments, or fragments of DNA at a time, and this in a reasonably rapid time frame, such that it now takes a much shorter amount of time and much less cost and resources to sequence an entire human genome, or genome from any species for that matter. This technology has also been used to then sequence the genomes of cancers, which are different from the sequences of the genomes of the humans from which these cancers have arisen, because they've got all these mutations in them. So now, over the course of time, thousands of cancers have been subjected to whole genome sequencing using this approach, and we've been able to learn much more about what is the landscape of gene mutations in cancer. Cancers can have a lot of mutations. They can have thousands of mutations in their genomes. And sometimes it's a case of how to separate the wheat from the chaff, as they say. Which of these mutations are important, which ones are driving the behavior of the cancer, and which ones are critical to the survival of the cancer, its ability to grow and spread. These mutations that seem to be the important ones, we call them driver mutations in cancer genomes. And so, a lot of work has gone into identifying not only what mutations there are in the DNA of cancers, but also which are the really important ones. And from there, treatments have been developed to try to target the effects of these driver mutations in order to treat cancer, as we have done with the case of Mr. Smith, giving him treatment to target the effects of his BRAF mutation in his melanoma. Okay. So that is the basic idea of molecularly targeted therapy. And just to go over the therapy part a little slower, let's say here's a cancer cell. And this cell is being driven by, say a BRAF protein. As in the case of Mr. Smith, here's the protein, this yellow protein. And let's say we want to inhibit this with a drug. So that is going to be the molecular target that we want to hit with our drug therapy. Because this molecular target is driving the behavior of the cancer, which includes its ability to replicate itself and grow into multiple cancer cells, which will grow into a tumor. Those cancer cells then have the ability to spread through the body and grow into other tumors. And we think that this protein, which is the product of a gene mutation, is the target that we want to hit with our drug. So someone develops a drug to treat this patient with. Here's a drug that we're going to apply to treat this cancer, like the drug treatment we gave Mr. Smith, for example. So this drug, the idea is we give it to a person and it's going to be absorbed into their system, circulate through their body, and hopefully reach all the cancer cells in the body, and then get to the target protein within the cancer cells that's being produced by this mutated gene, and block the driving effects of that overactive protein on the biology of the cancer. Okay, so if we can do that, if we can inhibit the function of this molecular target in the cancer cells, then hopefully the next thing that will happen is that we can kill those cancer cells, or at least limit their ability to survive, limit their ability to grow and spread, proliferate, and try to control the cancer for as long as we can this way. The hope, of course, is that we could cure a cancer this way. Okay, so that's kind of the Precision Oncology 101. When researchers, cancer scientists are trying to figure out what might make a good molecular target, these are some of the considerations. You want that target to be essential for cancer cell growth and survival. You want it to be a driver of that cancer. If the cancer can survive without the function of that molecular target, that gene or protein, then you're not going to be able to treat the cancer very effectively that way. So you want it to be essential for cancer cell growth and survival. You want this molecular target also to be selective. In other words, you'd like ideally that this target is found only in cancer cells. That's not always the case. Sometimes we have targets that we aim for that are found also in healthy cells, but maybe they're much more important to cancer cells for some reason. Maybe because of these types of DNA mutations, for example, that make those proteins different and more active in cancer cells than in healthy cells. So you want the target to be essential. You want it to be selective for cancer cells. There's a word that's not maybe really a word. You want the target to be druggable. In other words, you want to be able to develop a drug or some kind of treatment that you can use to specifically hit that molecular target in the cancer cells. So if you don't have that, then what are you going to do? Okay. And finally, you want this target to be measurable, so that you want to be able to identify this molecular target in an individual person's cancer, so that you can identify whether their cancer is going to respond to your targeted treatment. So those are some of the essential features of a good target for therapy. Okay. So this is some of the work that the Terry Fox Marathon of Hope Cancer Centers Network is doing, and some of you will be familiar with this. The great Terry Fox needs no introduction to most Canadians. And his family has carried on his legacy, raising funds for cancer research, really with the aim of trying to improve cancer treatment and to find cancer cures. One of the approaches that the Terry Fox organization has taken with this, the Terry Fox Foundation and their research funding arm, the Terry Fox Research Institute, is to get cancer researchers together across the country to form this network, which is known as the Marathon of Hope Cancer Centers Network. And this has been stitched together sort of region by region across the country. The first cancer centers to join this were the Princess Margaret Cancer Centre in Toronto and the BC Cancer Agency centered in Vancouver. And the Prairies since joined the consortium. Other cancer centers in Ontario are joining with Princess Margaret to form the consortium in Ontario, other centers in BC. Quebec has a consortium, and Atlantic Canada has a consortium. And we were the last group in, being the smallest. I guess they wanted to cover the country as quickly as they could, so they started with the big places and worked their way to us. But we've now been part of this network for a few years. So this is the Atlantic Cancer Consortium, part of the National Marathon of Hope Cancer Centers Network. And you can see that many institutions throughout the country are involved in this, including many throughout Atlantic Canada. The Terry Fox folks refer to this as the Team Canada of Cancer Research. We have so many of Canada's leading universities, cancer care centers, research centers across the country involved in this work, including several from our region. We also have to acknowledge the many partners that have contributed financial support to the Terry Fox Marathon of Hope Cancer Centers Network across the country. So, the Terry Fox Foundation has invested funding in this. The federal government has more recently invested quite heavily in this, And across the country, there are regions, throughout the country that have cancer research funding agencies and local hospital foundations, university foundations that have contributed to this effort. So a real national galvanizing effort, the kind of thing maybe we need these days more than ever. So the goals of this consortium, first of all, was to build this national network. The vision set out by the Terry Fox Research Institute is to build this network so that together we could conduct important and large-scale cancer research, the kinds of things that we couldn't do individually. Even the biggest flagship cancer research centers in the country, couldn't do on their own. And create power in that, and also to give the opportunity for people living with cancer from across the country to get involved in and to benefit from participating in the research, while building, and strengthening the capacity of cancer research across the country. The focus of the current project of this consortium is to build a large resource for cancer research comprised of the genome sequences of 15,000 cases of cancer from across Canada. This aims to be inclusive, diverse, covering as much of the country as possible, with as much diversity and inclusiveness in the cohort as possible. And there are committees and working groups that are part of the consortium to try to ensure that this is a diverse representation of the landscape of cancer in Canada. So this is what we've been really focused on as a national consortium for the last couple of years. This network has 42 member institutions from all 10 provinces. We have a patient partner committee at the national level with over 30 members. Some of you may be members. Several different working groups to drive this work forward on everything from understanding clinical oncology to genomics to patient perspective, on this research. We have over 600 individual members of the network across the country, including several early career cancer researchers who hopefully will be the future leaders of this network. So far, out of our goal to get the sequences from 15,000 cancers, we've done over 8,000, and hoping this year to make a big push to get the other 7 done. Now that the effort is sort of ramped up fully and is in full swing, we need to get the information about how those patients do. What is the stage of their cancer? How are they treated? What are their outcomes? And we're busy now gathering that information as the sequences get done. And we have a case that has successfully had its genome sequenced. Then we go find out what happened to that person with their consent, of course. Everyone signs a written informed consent to participate in this, and what's involved is explained to them. And so we collect their information and enter it anonymously, or in a de-identified fashion, I should say, into a large database. And this is going to be a very powerful resource for cancer research. This amount of genomic data on this large number of cancers, annotated by the information about how the patients actually did with their cancer, is going to help us gain new insights into the biology and treatment of cancer that I think the world will be interested in hearing about. And we've got a diversity of different types of cancer and different cohorts of cancer, people living with cancer represented. And we've had to build some infrastructure, to make this go as well. New tumor banks and new sequencing, capacity and that sort of thing. Okay. The Atlantic Cancer Consortium, is our 4 Atlantic provinces: Newfoundland, Labrador, Prince Edward Island, New Brunswick, Nova Scotia. I think, as Terry set off to run across the country back in 1980 from Newfoundland, and that the first leg of his journey was through Atlantic Canada, that it carries some real meaning to the Terry Fox Foundation and the Fox family that Atlantic Canada is fully engaged in this effort. And those folks are great about coming to visit us and encourage us to participate and to help us succeed, in line with Terry's dream that we find a cure for cancer. Our part of this national effort includes 9 institutions from all 4 Atlantic provinces. We have a dozen patient partners on our council. working groups representation from Atlantic Canada on most of the working groups. We have over 80 individual members from Atlantic Canada, including 10 early career researchers. And our commitment to the consortium has been to sequence just over 1,000 cancers from Atlantic Canada. And we're almost halfway through that, in line with the rest of the country. And we're just starting to collect the clinical information about how those patients did. And there's active research being done on this, with this resource in Atlantic Canada. So what do we hope to achieve? I think in general, we want an improved understanding of cancer biology. Hopefully we'll uncover new things about cancer genomics that we didn't know before and understand what that means for the biology of cancer. And hopefully we can use that information to develop identify new targets for cancer therapy and to develop new therapies to treat cancer. And, this is all the kind of work that is forward thinking and will benefit people with cancer in the future. But, there's always a sense of urgency to this. There are people today who need some advances made. And, we're hoping that, we can advance the research of this network to the point where it can also provide more tangible and more immediate benefit to people living with cancer who participate in the research. Certainly a big part of participation in research like this is understanding that a lot of the benefit might be for people in the future who walk behind us through this experience with cancer. We'd like to try and apply the benefits of this research as soon as we can. So one of the things that we're doing in Atlantic Canada to try to achieve this is we're partnering with the British Columbia folks, the BC Cancer Group, who for the last decade or more have had a program called Personalized Oncogenomics, or POG, POG. And this diagram is from the BC group, from one of their publications of the work they've been doing over the last number of years, illustrating their process of trying to bring precision medicine to patients as quickly as possible. The idea is, if you look at step one, you're enrolling a patient in the program. A sample of tumor is obtained through a biopsy. That material is then subjected to next generation sequencing. So we want to look at all the DNA and also the RNA, another form of genetic material, in these cancer cells, and collect that information into a repository, and then analyze that information for that individual person with cancer, understand their cancer's genome, discuss that at what's called a molecular tumor board, where there are both clinical and scientific experts that can look at this information and try and interpret it and say, okay, what does the genome of this person's cancer tell us about what treatment possibilities could exist for this person sitting in front of us, that maybe we wouldn't have otherwise thought of. What's their cancer's genome telling us about how we should treat their cancer? We might also learn something when we're looking at genetic information about whether or not that person does in fact have an inherited predisposition to cancer that we didn't realize before. Or maybe it helps us to understand an inherited predisposition that was evident from their family history. And we need to be able to deal with that. We need to have appropriate genetic counseling available for people for whom such findings are made. But for that person sitting in front of us who's faced with cancer now, some of these people are running out of options. What can we do? What does their cancer's genome tell us that we could do to try to treat their cancer? And so this is where hopefully this molecular tumor board recommends some actions to the treating oncologist that can be considered in terms of how to treat that patient. Sometimes it means we should reach for an old-fashioned chemotherapy drug that we never would have thought would work for a person with their type of cancer, because for most people it wouldn't. But maybe the genome of that person's cancer tells us that we should use that old-fashioned chemotherapy drug, and it actually might work pretty well. Sometimes it tells us we need to use a more elegant sort of molecular targeted therapy like we did for Mr. Smith. And maybe by doing it this way, we will uncover molecular targets that we don't normally uncover in our clinical lab now. I'll just emphasize that Mr. Smith was treated in a very standard of care type of way, shall we say. His approach was not research. We do implement, precision oncology in regular practice now, but we're much more limited in what we can do there. We looked for a BRAF mutation in Mr. Smith because we know that almost, maybe half or even more than half of melanomas have that type of mutation. But in this type of approach, you might be finding mutations that you didn't expect in an individual person that might tell you something about how to treat their cancer that you wouldn't have thought of before. And there are lots of challenges with that. I mean, sometimes the treatment you would like to give just isn't available to that patient. But if you can find a way, then you might be able to treat this patient based on the results of sequencing their cancer genome and see what happens. And so what we hope to do in Atlantic Canada here is we're going to start out by sending some of the sequencing data that we've already got from the ongoing sequencing project to get 15,000 cancer genomes done across Canada. And so we're, I treat multiple myeloma, which is not melanoma. It's another kind of cancer. So I'm confusing things by talking about two cancers with similar names. But we have a bank of multiple myeloma specimens in our research lab in St. John, New Brunswick. And so we've sent a few of our cases that have been sequenced at the Atlantic Cancer Research Institute in Moncton, and whose data have been uploaded into the Atlantic Data Repository in Newfoundland. And a colleague in Nova Scotia, who's our head bioinformatician for the network, is sending that data off to British Columbia. So you can see this is a real interprovincial effort, both in Atlantic Canada and across the country. The folks in BC are going to run our data along with some clinical information that we're providing to them about these patients through this process and tell us if they think there are any new treatment options we could consider for these patients in St. John. So this is a very small pilot just to prove the principle that this sort of thing could be done in Atlantic Canada, and hopefully helps us to iron out some of the processes and to help us start to think about how we could do this, wholly in Atlantic Canada, so that we have a responsive local approach to enhancing our ability to deliver precision oncology in a research setting, in Atlantic Canada. These folks in BC have published a couple of years ago their experience. At that time, over 700 people had taken part, and of those 722 participants, they were able to get complete genetic sequencing of tumors from 570 of the participants. And remarkably, they found that about over 80% of those 570 participants were found to have information in their cancer genomes that could potentially inform the way those patients could be treated, and varying degrees of ability to then apply that information depending on the treatments that were available to that patient at the time. So to summarize that, they found that on the left-hand graph here that out of these 570 patients, that there were over 100 who received some type of standard therapy that was directed, a therapy that was routinely available, that was selected for that patient based on their cancer genome, and that over half of those patients seemed to benefit from that treatment. Some of these patients were able to be directed into a research study of a new therapy, a clinical trial based on their cancer genome results. And again, of those folks, over half of them seem to be benefiting from that. And then some folks were able to be treated what's called off-label therapy, so treatment that might not have been routinely available for their particular type of cancer. Maybe there was a BRAF inhibitor given to a patient with, a different type of cancer that had a BRAF mutation besides a melanoma, for which this treatment wasn't routinely available, and some of those patients were able to benefit as well. So they've had a positive experience. It's certainly a select subset of cancer patients in BC that have been able to access and benefit from this approach, but certainly they can highlight some individual stories of really striking outcomes from this approach, including the use of some treatments that you would never think would be cancer treatments. I mean, they've got some great stories about success treating people with advanced cancer with blood pressure pills, because the cancer genome suggested that this might be effective, and indeed it turned out to be. So really really insightful information that sometimes is gained. Not to suggest that this is necessarily the approach that all cancer patients should be taking today. You know, we have very effective treatments for certain kinds of cancer already, some of which do not require, you know, cancer genome sequencing, you know, to be effective or to access the therapy. And we know that this doesn't work for everybody. There are people who can have their cancer genome sequenced and, you know, we don't have any action we can take as a result. And there's a lot of people like that still. So this is not the solution for everybody. This is not to sit here and say this is the cure for cancer, but it's moving things forward. And I think, you know, if you look back at cancer care over the last 50 or 60 years, I would say that progress has been stepwise. You know, we have not had that one big, oh, we've got the cure for all cancer. You know, instead, what we've learned over many years and decades is that cancer is many different diseases, and some of this, what distinguishes individual cancers from one another, can be identified at the genetic or genomic level like this, and this can give us insight into how to make progress and move forward. You know, so that maybe a guy like Mr. Smith can live an extra 5 years that he wouldn't have otherwise been able to do. And that maybe in the future, a guy like Mr. Smith maybe will live an extra 20 years or 30 years, you know, if we can do this even better in the future. So that's the idea. And so what we hope to get back from our myeloma pilot project is reports like this that can be reviewed potentially by a clinician who's treating a patient with myeloma to say, okay, what's all the genetic information that's being uncovered here? And what does it tell us about how we could potentially treat this patient? And one of the things that the folks in BC have come up with is a way of trying to automate this analysis to a certain extent so that this huge amount of genomic data on an individual patient's tumor can be analyzed and digested for clinicians to look at and say, okay, what are the treatment options that might be found in this tumor's genome? So we're working on this, and we're working on implementing this platform for analysis of cancer genomes in Atlantic Canada. We are nearing the end of our current 3-year cycle of funding from the various funders of the Marathon of Hope Cancer Centers Network. And so in the next year, our focus is on sequencing those 15,000 cancer genomes from across Canada and creating this immensely valuable resource for cancer research. We are thinking ahead to where we want to take things from here as a national network. And one of the leading ideas that's emerging is that this kind of thing where we take the example of the British Columbia Personalized Oncogenomic Protocol, similar protocols that have been running at the Princess Margaret Cancer Centre for some time as well in Toronto and others across the country. There's some in Quebec, certainly in Montreal. You know, roll this out more across the country so that more of this kind of whole genome cancer sequencing information can be returned to the clinicians who are managing these patients to see if there's information in there that they can potentially use as part of a research protocol. And so there's a protocol that's being developed that's currently got the name Generational, although it looks like Generation All, and that's very intentional. The idea being to gather information that will benefit, you know, cancer patients in Canada and elsewhere for generations, and that the center of this generational project is going to be to try to go ahead and expand on this ability to return the results of cancer genome sequencing to the treating clinicians, doctors, and oncologists, and those who are caring for the patients. So we're hoping to be one of the first participants here in St. John to get this protocol activated, and I'm going to be speaking about this with colleagues across Atlantic Canada. I actually haven't had the opportunity to speak to them too much about it before I've had the opportunity to speak to you about it. But we want to try and start piloting this so that we are in a good position to talk to our funders and supporters and say, hey, this is where we're heading with this, and you know, we could use your support to roll this out. Okay, so so that's where we're hoping to go, and these are ideas right now. in their early stages, and we're just trying to do some initial work to lay the foundation for expanding on the great things that have happened through programs like those in BC and Toronto for the rest of the country, and to build our capacity as a country to do this. So I'd just like to conclude my slide presentation by thanking you for your attention and thanking you for your engagement and support of cancer research. You know, this is really a team effort. It requires researchers, clinicians, people who have been diagnosed with cancer and are living with cancer, people who believe in the importance of cancer research and can support it financially and in other ways. The institutions that we work with here across the way, there is the St. John Regional Hospital where our cancer clinic is, and this is some of our cancer research team in St. John here at on the university campus next to the hospital, where our research facilities, some of our research facilities are, as well as in the hospital. And this was a great day when we had Fred Fox come and visit us and inspire our team to keep working hard at this together with the rest of the country. So I'll stop there, and thank you for your attention, and that'll give us a little bit of time for discussion. And I don't know if someone wants to moderate that discussion. I know there's been some things flying through the chat while I've been talking that I haven't stopped to answer. I thought maybe we could do some of that at the end. So I'm going to stop sharing my slides, and I don't know. I suppose I can do this myself, but
no, I'm right here, Dr.
well, thank you, Jennifer.
Thank you so much for an amazing presentation. We do actually have a couple really great questions for you. Would this type of precision oncology genome sequencing also apply to the BRAF V600E gene?
Right. So in fact, so someone is very knowledgeable about this BRAF mutation. And this V600E mutation, this is a notation that refers to the most commonly seen mutation in this BRAF gene in cancer in general and in melanoma in particular. And in fact, Mr. Smith had a BRAF V600E mutation, and he was treated with this therapy. I will say, though, that if you are, wondering maybe why you're not receiving this therapy or haven't received this therapy, if you or your loved one has a V600E mutation melanoma, it's because we actually have other treatments that we often try first because they may turn out to be better in the long run than even what we gave Mr. Smith, who started his treatment a few years ago. This treatment often is now a second-line therapy for people with BRAF mutations in their melanoma, and we typically start with immune therapy, but that's a talk to your oncologist type of a question.
Yeah, no, I completely understand because I'm actually a melanoma patient myself, and that's the first line of defense was doing the immunotherapy for melanoma here in Newfoundland. And that just came
Sounds
out.
very, very much like what we would do in New Brunswick.
Yeah, so and that
world,
one
really.
basically came out in about 2017, I think, because they said to me when I was diagnosed in 2019, had I been 2 years earlier, it would have been an automatic death sentence for me for the type of melanoma I had. So, precision oncology is definitely doing great work. Another great question that we have, because we only have a couple minutes left. When a tumor is tested, how many mutations is it tested for?
Well, there's a couple of different scenarios there. In the clinical setting, in hospital labs across the country that are doing this on a daily basis for patients, we use what are called sequencing panels, which is a way of using next generation sequencing to look for a subset of mutations that we know happen on a recurring basis in cancers, and that we have treatments potentially that we can use to target those mutations. So we started out doing that with older techniques, and we would only maybe test for a few different mutations, a handful, 5, 6, 10, 15 in a given tumor. And then everyone started moving to larger panels. There were some efficiencies in testing all the cancers for 50 mutations, or maybe mutations in 50 genes, I would say. You could test for multiple mutations in 50 different genes. Now we can do 500 genes with these clinical panels. And so that's a moving target. It's an expanding number. that's being done in the clinical setting. With the approach that the Marathon Hope Cancer Centers Network is taking, the idea is to basically sequence the entire genome. And so you're going to get a huge amount of information, including you're going to detect a huge number of mutations. There's typically thousands of mutations detected in a given tumor this way. And then the challenge is to make sense of it. But this is where, there's a leap forward in going from what we do now in the clinical setting on a daily basis to what we could do if we translate this research approach to patient care.
Right. And one final question that will pretty much be the end, so we don't run over too long. But this one's brought up a couple of times, and I think it's a great way to end our conversation today, because we all know we could go and talk for hours, because this is so amazing. But how would one apply to have their genomes included into these sequencing projects?
Great question. So right now, in the interest of making progress as quickly as we can, the emphasis has been on sequencing tumors that we've already got in hand. The tumor tissue has to be processed in a certain way fairly quickly and stored in a certain way in order for the tissue to be suitable for this type of whole genome DNA and RNA sequencing approach. And so not every case of cancer is there that material available to do the sequencing. So to put this 15,000 case cohort together, we're concentrating on sequencing basically cases we've got in hand that we know we can do it. Going forward, the hope is that eventually, when we have this generational protocol really up and running, which is going to take a fair bit of time, so it may be a couple of years before it's widely available for people to just say, hey, I want my tumor to be sequenced, or my oncologist wants my tumor to be sequenced. But if you're in BC or if you're in Toronto, you might have the opportunity to participate in the research protocols they've got going already. Occasionally we will refer patients to centers that have this up and running if possible. Sometimes there can be a cost to that, et cetera. But I would say stay tuned, and it's not something we can offer you today, but that this is the goal in the next couple of years, is to have something like this more widely available in Canada.
Well, thank you so much. You did an amazing presentation and really kind of opening our eyes to this big word that we're throwing around of precision oncology. And you really broke it down for us to what it actually means and where it's going in the future. So I really want to say a very big thank you and appreciate your time and your expertise in this subject. And we will conclude the session. And we just want to say, again, a very big thank you for all your knowledge on this topic.
Well, it's an honor and a privilege to be here to talk about it with this group today of very interested people. I can see that we only covered a small fraction of the chat, but I hope that if we didn't get all your questions answered, at least everybody got a little something maybe out of it.
Definitely. Well, thank you so much, and you have a great rest of your day.
Thanks very much, you too.